Hey, it's Mr. Chemist. Here's your Friday lecture for biox. We are still working on evolution, and you should have just taken the quiz already. And then we'll head over to your lesson today. So if you look at the schedule for today, after the quiz, it's 2 sets of notes, one on how we get different species, and then one on how we organize species into a chart. We call that clades or cladistics. And then there's a little bit of homework. If you need help on review on that one, called the cladistic activity review, that's something we can talk about on Monday or Tuesday. And that's really going to help you make sure you can do those, because we have a project about lizards and you'll be building clades and cladograms with that. The homework that's due next week is here, reading and how flowers change the world. And so you can start working on that. If you need help too, the Amoeba sisters explain these mechanisms of speciations pretty well, and it's brief. It's only like 7 minutes. Yeah, you can say hi.
What's good?
Hey, it's Ben. All right, well, let's head over to the notes here. It's been a while since I've done a Zoom. It's kind of fun. So there are different ways that we can look at evolution, macro and microevolution. Macro is like these larger speciation events. Microevolution is kind of looking at differences in the DNA, like small changes in an allele or in the gene. So today we really want to talk about those big changes when we get new species, where there's been a lot of changes in the DNA, a lot of changes in the allele. It also includes some of the extinction events. This is important for evolution because then there's room for new organisms to branch into new environments when some organisms die out, like from a meteor or volcano or climate change or something like that. And then at the bottom here, we have the definition for speciation. It's basically, if you would take the word species and make it into a verb, this would be it, making a new species, speciation. This is the verb. All right, let's define species. There's a couple of definitions for species, but this is the good one for biological species. A group or population whose members have the potential to breed and produce fertile offspring. So not only can they breed, but their offspring will be fertile to be able to have children. So that would be the biological definition. That's a pretty important one for you there. There's a couple other definitions, and your book talks about those too. But I think this is probably the best one. A counterexample would be the eastern and western meadowlark. These live in the United States, and they're similar species. They look very similar. They could breed together, but they don't. And they're actually isolated by song. They have different songs, and so the females will not be attracted to the males with a different song, even though their territory does overlap a little bit in the southern part of America. And remember, like we talked about earlier this week, there's always diversity within one species or variation within one species. Without the variations, we wouldn't be able to have evolution occur. Your book talks about these 2 different patterns for evolution, branching and non-branching. We're really going to focus on the branching ones. So in non-branching evolution, an organism can change over time and a new species would not form. But if 2 populations of the same species are separated, they could branch and split into 2 different species during a speciation event. I think too, this one's a little misleading. because over time, some species won't actually change. They will stay stable. They're well adapted to their environment. Their environment hasn't changed. So be careful with this non-branching one as well. All right, let's talk about how mechanisms of speciation here. There's two big types, allopatric and sympatric. Basically, we need a group of organisms to be isolated in some way. And the most easiest one is geographic isolation. Maybe there's a mountain range. Maybe there's an ocean. Maybe there's the Grand Canyon. There's different squirrels on different sides of the Grand Canyon. They're different species. They're separated by the canyon. It's huge. Or species could form in the same area. They're not separated by geography. Maybe there's differences in their DNA. Maybe they mate at the different times of year. Maybe they have different behaviors, like the songbirds. They could live in the same area, but they have different song or different mating dance. This would be an example of sympatric speciation. They live in the same area, but they are still going to produce a new species. So here's your book. It has examples of these with trees. Allopatric, what if there's a river or a canyon where the tree pollen can't get across? It'd probably need to be a much bigger canyon than this, as shown here. And then sympatric speciation, maybe a group of trees are starting to breed a little bit earlier than some other ones. And so their pollen is not viable by the time the other ones become fertile. And if that happened over many 10, 50, hundreds of generations, maybe there could be enough genetic changes there that there could become a new species in the same area, the sympatric speciation. Okay, this is a pretty good summary. This diagram, I like it, but it is a little bit more complicated. So if there's individuals of different species, when can look at all these possible places where they could have barriers to breeding. These are before the sperm and egg join, before fertilization, before the zygote forms. This is during the mating attempt, and then this is after the zygote forms, or after the baby is born even. And if all of these, it usually only takes one of these to be a problem, a barrier for populations to not breed. But if there are no barriers, then you do get viable fertile offspring at the end. Make sure you know pre-zygotic and post-zygotic. Pre means before the joining of sperm and egg, before the fertilization and mating, and post-zygotic would be after. It affects the children or the children later in their lives when they are adults trying to mate. The book does a pretty good job of getting examples of this one. Temporal isolation is separated by time. They breed at different times of the year. Habitat isolation, look carefully behind the snakes. This one lives in water. If this one lives mostly on the land. Yeah, they're not going to meet. They live in different habitats. These are from the Galapagos Islands here. They have a different mating dance, a courting ritual. And then this one, I had to look at the picture again and read the caption. Did you catch this one? These snails' shells spiral in opposite direction. One goes this way, one goes this way. And they, because of this, their reproductive organs can't line up, and so they physically cannot mate, which is kind of interesting. And then this last one, gametic isolation. What if the gametes, like the outside coating of the gamete cells, just physically they don't match? We would have a bunch. It's not possible for the sperm and egg to join. So that happens with these different red and purple sea urchins. All right, let's talk about post-zygotic barriers then. So this would be after the baby is born. What's going to be not the best with the resulting offspring? So the first one is reduced hybrid viability. This is when the offspring really aren't the healthiest. They're frail, maybe they don't fully develop. An example here would be this salamander hybrid. And then for horses, if you cross a donkey and a horse, you get a mule. Well, you can't take two mules and have them mate and get a baby mule. You have to, they're not fertile. Okay? They're not fertile. You'd have to go back and get a horse and a donkey and mate them together. So this is called when the offspring of the mix, the offspring of the hybrid can't have babies, it's called reduced hybrid fertility. And then when you have a little bit of both, that's called hybrid breakdown. The rice hybrids at the left and right are fertile, but then farther down after they breed again, the next generation are smaller and sterile and not viable. So they're not viable and they're not fertile like 2 or 3 or 4 or 5 generations later. That's called hybrid breakdown. Maybe you guys watch like Tiger King or something. They definitely show how inappropriately when the tigers and the lions were mixed and you get a liger, similar to like a horse, a donkey, and a mule. This is not the best mix. They have quite a bit reduced viability. It's not the healthiest animal. It's quite a few animal activists would argue that that's pretty cruel as well. So overall, we need speciation to occur. We need a separation of reproductive, and it could be geographic, like a river, a canyon, ocean, a mountain, or it could be some of these like cellular and behavioral and reproductive barriers, both pre and post-zygotic. You can watch this video from the notes if you want. It's pretty decent, actually. The Birds of Paradise project. I know a lot of you guys watched some of that stuff in 7th grade. You can try to talk this over with a partner, or you could write in your journal about it. And the last idea here is how fast does speciation occur? There's kind of 2 conflicting theories here, and probably depending on the case, they have different. Either one could be true depending on the case. The gradual model is there's lots of small changes occur by big changes. The speciation occur by lots of tiny little small accumulation of changes over time, which is shown by the bottom one here. These 2 populations of butterfly could have small changes, and over time there could be enough where they can't breed anymore, and they would be individual species. And then contrast that with punctuated equilibrium. This is where there's rapid change in the environment. It could cause the speciation to occur much more rapidly because, okay, let's think about it. If the environment changes and the organism either survives or doesn't, so there might be some rapid adaptations and speciations there. So that would be punctuated equilibrium. This could happen after like a natural disaster or some big change in the environment. Okay, I think that's it for that one. Let's go over to the second one. You guys are ridiculous.
What's up, Tyler? And for now. And
yeah, you do stop by. So they can block once in a while.
Who else?
All right, here is the clade note. So let's talk about how do we organize living things over time, and how do we put them into a chart to show how they're related and how long ago they had a common ancestor. So let's take a look at these. There's a little bit of review from the speciation stuff here. And then we really want to focus on this right type diagram here of how to organize living things. So for this one, let's see here. What is that? So there's quite a few variations of this word. Clade, cladogram, cladistics. So make sure you can use all of theirs. The cladistics is kind of like the study of. The cladogram is the diagram. And then the clade is the group of organisms that share that trait. We'll take a look at those together. Well, here you can see, let's define all those words a little bit more. Okay, cladistics is the evolutionary history for a group of species. So this is the study of how we organize living things. Here you can see the modern organisms, A, B, C, and D, and some of the traits that have been derived over time. One would be present in A, B, C, and D. The second trait would be present just in B, C, and D. This third trait and everything above it would be shown here in C and D. And this fourth trait is unique to this character here. And so typically we can do these with the fossil record. We can look at living species, we can look at extinct ones, we can do DNA studies, and then we can build this branch tree. There are several different styles of this tree as well. Some go from bottom to top, left to right, it doesn't really matter. Some have horizontal branches, some have vertical, some have these where they just split. And we'll see examples of those. Okay, so the actual diagram is called a cladogram. So once you get this made at the end, it's called a cladogram. And then all the group that share a common ancestor is called a clade. Okay, a clade. So here's the clade of the different plants here. What is this? Felicinophyta and Bryophyta here, the moss and liverworts, and then we have the ferns, Felicinophyta, and then the most modernly derived plants here. We have the Coniferae, which you might have heard of as conifers, and then the Angiospermophyta or angiosperms. That's all the flowering organisms, the plants, and all these would be in the plant kingdom. They would all share traits of plants. And as new traits are derived, they would branch into new groups here, new clades. Here's one from animals. Derived characters are traits shared by different degrees of clade members. So let's look at these. Here's the modern organisms. We have amphibians, we have elephants and rabbits. We have some reptiles and snakes. Okay, and you can see the different groups of organisms here and how they're related. And then why would we organize these this way? It's a little different. We're gonna look at these based on their shared derived characters. So if we go back all the way off this chart, there's an ancient organism that has 4 limbs with digits. And all of these modern organisms share that trait. They're in that clade. And so if you look at this tan area, that would be the tetrapoda clade. Tetrapod, 4 feet. The next trait to develop was embryonic and amniotic fluid. So here's the next derived character. So all of these organisms after that would include that trait. So everything in blue. Skull opening behind the eye would be the next derived trait. So yellow, green, and red would all have those, etc. A node represents the most recent common ancestor. So that's not very clear on this diagram. The node is where these 2 lines join. So if we go back to all the most recent common ancestor for all of the tetrapods, it's going to be this node right here. Okay. How about the node for all of the all of the organisms that have an opening in the side of the skull. That's going to be this node here. Okay, everything after that. So here you can see how to read a diagram with clades. Yeah, the most recent common ancestor would be here. And then here's the node where they would have that common ancestor. And then they would split depending on new species or new traits. Here would be the living relatives of those common ancestors, the descendants. Branches on the tree usually represent a speciation event, or they could represent larger groups. And then after that node, after that where the line split, they're gonna have a unique evolutionary history. Okay. And then how about a common ancestor? We see those at the node. What is the only thing A and B have in common with? It's just gonna be everything above here they would have in common. All right, how about clades? This sounds like a good test on a BioX test question. A grouping that includes common ancestor and all of the descendants living and extinct. So this has a node here and everything after it. This one is after node and how it's evolved separately. It can still technically be a clade. This one's not a clade. It has everything after this node, but this one, they don't share a common ancestor in this red square. That's not a clade. Same with this one. They do have that node, but they're missing this one. This one needs to be included to be a full clade. There's a couple different places for you guys to practice in here. How are these organisms related? Which ones are more closely related? Could you draw a little cladogram and show those nodes and have them branch? How about these organisms? Could you organize them into branching, which is more closely related? So pause the video, try this out in your notebook or your journal. And here's some possible ways to organize those. You look at the different traits or derived characteristics and to find out which ones are more or less related to each other. And here you can see the different styles of cladograms as well. Here's the one we were using with the just splits lines. Here's the one with the speciation event. It has a larger node. It has that full big horizontal line to really emphasize that. And I think there's one more thing I wanted to show you down here. Yeah, Tiktaalik that we saw in the inner fish. Here you can see this ancestor, ancient ancestor of Tiktaalik and all modern land animals here. And I do like this misconception down here. Taxa that are near the bottom of the left-hand side evolved earlier than those. Not necessarily so. What it does say is when did they have a common ancestor? Well, things could definitely evolve afterwards. There could be splits up here as well. So here's some other cladograms you could look at. Look at the characteristics. Where do they share? What organisms share them? And which organisms are more closely related than the others? Sometimes too, they will pivot around these nodes. So if we took these two and flipped it with these two, it's going to be equivalent, which is a little confusing to me. I'm not sure why they do that. It's not the best notation. Here's another different form where we have more of the branching tree of life. You can see the different branches there. This one's based on genetics. Okay, that is the end of your second notes there. We were going to use something like this to do your project on Monday and Tuesday. And you guys will have a complex problem to solve for that lizard's evolution diagram, which we'll do on Monday and Tuesday. And we'll introduce that during class, and then you can start working on it. As far as today goes, you can watch the Amoeba Sisters over here. They're pretty good with speciation. They explain that first notes, especially allopatric and sympatric speciation. You can do this reading here, objectives 15 through 17. A little bit of review there. Derived traits, homologies, homologous and analogous structures, and clades and cladograms. Make sure you can follow that up in the textbook. Start reading this, How Flowers Change the World. It's a little bit longer reading, 5 or 6 pages. There's some questions that go with that one. It's pretty fascinating. 70% of all plants in modern times are flowering. They're angiospermophyta, and really how that has changed not only plants but the animals around them. It's pretty cool. That one's not due till Wednesday, so if you have a busy weekend, you could do that next week as well. But we also have this project next week, and then the test will be on Friday the 14th before Martin Luther King weekend. And then the lizard project will be due after Martin Luther King weekend. All right, well, that's about it for Mr. Chemist. If you have anything that you need help on, you can send me an email, or we'll see you on Monday. You can also always check the modules here. That's a great place to see what is due and how many points different things are. So anyway, it's nice to chat with you, and I hope this is useful for some of you guys. So have a good day.